Okay, hello everyone. Sorry for making this video so late, but today we're going to be covering activity 4 in exercise 3, biological molecules, and this activity is going to be discussing lipids. Let me go ahead and share the PowerPoint real quick. Okay, so lipids contain the same elements as carbohydrates, but with fewer oxygen atoms. So as you will see, lipids have hydrogen, carbon, and oxygen elements, except they're going to have a lot less oxygen atoms. Unlike the other biological molecules that we will be discussing, lipids do not have true monomers. So as we saw with carbohydrates, the monomer was monosaccharides, but lipids do not have these true monomers. They typically have a carboxyl group as their functional group, and most of them would have this glycerol backbone, but we don't think of them as true monomers. So there are various forms of lipids that we will be discussing. We will talk about fatty acids. So fatty acid, an example of fatty acid is shown here. And a fatty acid consists of a carboxyl group, which is shown here. And if you remember, the carboxylic acid is one of the functional groups that was extremely polar. And in this case, it's different though, because this carboxyl group is not going to be polar. So when we discuss proteins, you're going to see that that carboxyl group is indeed polar. But here, this carboxyl group will undergo a dehydration reaction with the hydrogen up here from the glycerol backbone. And it will be nonpolar. So these fatty acid tails are considered nonpolar. So like I was saying, this here is a fatty acid tail. It consists of a carboxyl group, which is shown here, and then it has these carbon chains, as you can see here. An example of this would be butyric acid. Another type of lipid we're going to discuss is triglycerides. So triglycerides consist of a glycerol backbone. So up here we have a glycerol backbone, and it's joined with 3 fatty acid molecules by dehydration reactions. So this glycerol backbone will link up with the fatty acid tail, and it will do this at 3 spots. It will do it here, here, and here. So essentially what you're going to see is that this hydrogen and this hydroxyl group will form a water molecule, and this fatty acid tail will link to this glycerol backbone. Like I said, it will happen at these 3 locations, and this will form a triglyceride. So when you go to the hospital and you get your blood drawn, typically they measure your triglyceride levels, and triglycerides are found in your adipose tissue. Another lipid we will be discussing is phospholipids. So phospholipids are found in the cell or plasma membrane. So phospholipids have these fatty acid tails that are nonpolar. These are hydrophobic tails or water-fearing tails. And then it has a polar head, which is considered a hydrophilic head. So it is water-loving. So the hydrophilic head has a glycerol molecule as well as a phosphate group, and this phosphate group is charged. So the head is polar, the tails are nonpolar. And because of this, phospholipids tend to orient themselves in this manner where the heads are on the outside and the tails are interacting with each other, and they typically form these phospholipid bilayers. This is found in a cell membrane, for example. Another thing I just want to highlight very briefly is that, as you can see in these illustrations, that one fatty acid tail is shown straight. This is what we call a saturated fatty acid tail, and one has a kink. This is what we call an unsaturated fatty acid tail. And what you will see in phospholipid bilayers is you can have a mix. You could have 2 saturated fatty acid tails, 2 unsaturated fatty acid tails, or one of each, as shown here. And this is important because when all these tails are completely straight, this phospholipid bilayer is going to be very compact, and it's going to be very difficult for anything to come in and out of the cell. When you have an composition as shown here where one fatty acid tail is straight and the other one has a kink, this allows more fluidity in the phospholipid bilayer, so molecules or resources can move in and out of the cell more readily. So these are two other examples of lipids that we will not be discussing here, but if you want to read more about it, you are welcome to. So if we were in lab, you would be building lipid molecules like you did with the carbohydrate activity. So a typical fat or triglyceride consists of a glycerol molecule joined with 3 fatty acid molecules. Fatty acids consist of a carboxyl group and carbon chains varying in length from few to many carbons. So here we have 2 examples of fatty acids. And if you were in lab, the first activity you would be doing is building a butyric acid molecule. To do this, you would need a carboxyl group, because remember, fatty acids consist of a carboxyl group. So you would grab your carbon, 2 oxygens, and your hydrogen. And then you can see this is a double bond, and these are single bonds. And then you would build a carbon chain. So your carbon chain would consist of your carbons and your hydrogens. You would connect these to build your fatty acid. So here you build butyric acid. So if you wanted to build a triglyceride, you would have to connect these fatty acids to the glycerol using dehydration synthesis or dehydration reactions. So on our left here, we have our glycerol backbone. On our right, we have our fatty acid. So in order to join these two together, we need to undergo a dehydration reaction. So one hydrogen will be lost on the glycerol backbone, and a hydroxyl group, so a oxygen and hydrogen, will be lost on the fatty acid. And these will be able to connect as shown here. And when these 2 molecules join or link together, a water molecule is formed in the process. So if you were to connect 3 fatty acid tails to the glycerol backbone, you would form a triglyceride. So this is just showing that again. So here's your glycerol backbone in the salmon color. Your fatty acid tails are shown in yellow. And to connect the fatty acid tail to the glycerol backbone, you have to undergo a dehydration reaction. A hydrogen will be lost on the glycerol backbone, hydroxyl group on the fatty acid tail. The fatty acid tail will be able to link up to the glycerol backbone, and a water molecule will be formed in this process. The thing I want to discuss in this slide is the difference between saturated and unsaturated fatty acids. So as you can see on the top here, we have a saturated fatty acid tail, saturated fatty acid tail, and saturated fatty acid tail. So these saturated fatty acid tails are straight. You do not see these kinks. And a saturated fatty acid tail has a maximum hydrogens and no double bonds. So I think that it's pretty easy to see it here, but if you go back to our discussion about chemical bonding, we learned that carbons have 6 electrons. 2 of those electrons will be in its inner shell, and 4 of those will be in the second shell. And if you remember, your carbon's going to be happy when its outer shell has 8 electrons. That's when it's satisfied. So right now it has 4 electrons and 4 empty parking spots. So this carbon gets those 4 empty parking spots filled by this hydrogen, this carbon, this hydrogen, this carbon. Same thing goes for this carbon here. Those 4 empty spots are going to be completely saturated. They're going to be filled up by this hydrogen, this carbon, this hydrogen, this carbon. So in these saturated fatty acid tails, these carbons are essentially completely saturated. They are completely bonded and they are happy. What occurs in unsaturated fatty acid tails, or fatty acids, is that this carbon was supposed, was supposed to link up with the hydrogen here, and this carbon was supposed to link up with the hydrogen here. However, those hydrogens never showed up, it didn't happen, so instead a double bond was formed between these two carbons, and that creates these kinks. So double bonds in unsaturated fatty acetals create kinks in the tails, and this is also highlighted here for you. So I kind of also briefly talked about this when I was talking about the phospholipids, but saturated fatty acid tails tend to be very compact. So when you see this, you tend to see, or saturated fatty acid tails are found in hard fats, so like things like butter. So they're going to be very compact and they're going to be solid at room temperature. Unsaturated fatty acid tails have these kinks and they have more fluidity to them. And in room temperature, you will see that they are liquids. An example of this would be oil. So for lipids, you really need to know what their functional group are, like what functional group are found in fatty acids. That's your carboxyl group. You need to know the difference between saturated and unsaturated fatty acids. You need to know what elements are found in lipids. So you have your carbons, you have your hydrogens, and you have oxygen to a much less or not as many oxygens as we saw in carbohydrates though. And that's it for lipids. I will put up the video for proteins hopefully tomorrow. I'm going to split proteins up into two sections. So I'm going to first introduce proteins, and then I'm going to talk about the experiment you would be conducting if we were in class that has to do with enzymes. And then I'm going to just briefly probably talk about nucleic acids like in 2 slides because we have a full section of nucleic acids coming up. So I'm just going to give you the bare minimum. Don't forget you guys have a quiz due on Sunday, and there is no worksheet due this week. However, I do advise that you fill in all the blue boxes in your lab manual so you make sure that you understand these concepts. Okay, and that'll be it.